The reality of especially drug development business, in particular the field that we're in, which is pain research, the vast majority of the money and funding is in big pharma, and a lot of that big pharma is in the US. For ourselves, I mentioned we need financing to get through the IND phase. We also are going to need financing to get to the phase one clinical trials. We're talking tens of millions of dollars, and it's that kind of money isn't really available in Canada.
Hello, this is Dr. Karmy for Chronic Pain Chronicles, and today we have a guest with us, Dr. Chris Bladen from Zymedyne, a startup company which is developing a new medical treatment for chronic pain. Hi, Dr. Bladen. Thank you for joining us. I guess you introduced a little bit. How did you end up becoming interested in chronic pain, or was that even what you were interested in?
Yeah, specifically getting into pain research. I first started when I began my PhD in 2010 at the University of Calgary. Previous to that, I'd been working in biotech as an electrophysiology, and most of my work was examining ion channels in various other diseases, really, such as diabetes and cardiac diseases. But my focus on pain really started when I made it the subject of my PhD, which was to develop new therapies for pain.
So I don't know how these things work, but when I used to be involved, typically your supervisor has grants for certain things, and you don't really get to choose a topic. It's sort of given to you.
Yeah, I was in a unique situation in that I was working in Calgary as a electrophysiology for a startup company with my current co-founder, and they were looking at an epilepsy treatment actually. So that was what I was focused on, with the sort of ion channels and the biophysics of diseases, is that it's easily translatable across the spectrum of diseases. And so my supervisor then had grants for a variety of things, including pain. And when the time came and his company actually got merged and actually went IPO, they actually transferred to Harvard University. And I had a choice. I either went with the company to Harvard or I stayed behind in Calgary to do my PhD, or that was the offer anyway. And since I had a wife and young kids, I decided to stick in Calgary. I actually, believe it or not, did start looking at epilepsy in my PhD. But many of the same ion channels that are involved in epilepsy, because of the electrical activity, are also involved in the pain signaling. And I quickly became fascinated with the pain story. And it seemed like a much more direct avenue to what I was trying to do, which was I wanted my PhD to be more practical and sort of research focused. And so I pursued the pain angle. And right away, it produced a bunch of papers quickly. And when you're doing research and you're a student, that's really important. And so once the first one or two papers out, I really started to get this sort of idea and angle into looking into pain research and the role of ion channels.
So it sounds like the company was an outgrowth of your PhD thesis.
Indirectly, it was, although I worked with several other people in the lab that also worked on pain. Was actually one of those gentlemen that sort of discovered the idea. And I worked a little bit on that project as well. So it wasn't directly involved in anything I did in my PhD per se. I was working on actually developing drugs for direct targeting of the calcium channel 3.2, which is the focus of our research. And it was a few other people in the lab who had a focus on looking at indirect sort of regulation of the T-type calcium channel. And it was this research I had a pretty minor role in. I did experiments for them, minor role, that led to the discovery. And this was back in 2010, 2011, 2012. when this discovery was found, we felt that it was a big discovery. And so we did ask the university for help patenting the mechanism. But at that time, there was really no push to create a company from that. We just decided to make sure that we patented it before we went about publishing it.
So leave the door open for
To
future.
leave the door open in the future, which turned out to be a very, very smart move.
Which brings me to sort of the next stage, I suppose. The majority of PhD students, especially in Canada, don't start their own companies. In Stanford, yes, but in Canada, usually, I think academicians stay in academia. Very few make the jump to biotechnology company. Now, you did have a little bit of a background in biotech before you started your PhD. So what led to you jumping back into biotech?
Yeah, it's a funny story and it's quite serendipitous, really. Give you the earlier background story on technology, and it really sat there, patented in no man's land quite a few years. And indeed, as I finished my PhD, I was working in industry. I was working with a scientific company after my PhD, but I also was applying for academic positions. And in 2017, I got a tremendous offer from Australia, from Sydney, Macquarie University, to join them as a research fellow. And so I thought that was what I was going to do. That would be my academic pursuit. I was going to be an academic. And everything was going quite well with that. Still researching pain. That became my focus in Macquarie. But as everyone probably remembers, 2020, something came along called COVID. And because I was not an Australian citizen, Australia was very strict about its quarantine rules. I came home for a 2-week visit to my family in March of 2020. And at that point, the Australian government closed the door and I couldn't get. Yeah, so I really felt, as many did, that I would be losing my job position because I was unable to right away anyway do research. And so I contacted my old PhD supervisor and sort of asked him if there was anything happening in Calgary that I could work on or work with. And he suggested at that time the university had started this startup program and had some money for that. So he suggested I apply for it. So I did apply for it based on that technology that I talked about earlier. And we got the grant money, and the grant money allowed us to form the startup company. Basically, from those humble beginnings, we've grown. Turned out that I didn't lose my job in Macquarie. I was still locked up for 2.5 years, but I was able to carry on teaching and do research at the same time, fostering the company and developing it, and helping to get more grants to do the research. And it's really grew from strength to strength. And me and my co-founder just were able to get significant funding for it from the Alberta government to take the technology forward at a much faster rate than if it had stayed in an academic setting.
Okay, so let's maybe go back and discuss in a little bit more detail the actual idea behind the company. So it's a voltage-gated calcium channel type 3.2 that you are trying to interfere with. And then there's also an enzyme. So you don't seem to interfere with it directly, but rather indirectly through an enzyme, USP5. But I could be getting it wrong, so why don't you? Okay, correct.
Yeah, so in the research lab, when I was doing my PhD, it discovered that the 3.2 calcium channel is very important mediator of pain signaling. There are many proteins, of course, involved in the pain signaling process. Ion channels play quite a large part in the signaling, where the pain signal gets transferred from the sort of site of injury all the way up to the brain. And Cav 3.2, the calcium channel, is quite important in mediating that signal. And what the lab had discovered is that the increased activity of the Cav 3.2 was enhanced in the various pain conditions. And that when you block that channel, either directly or indirectly by inhibiting them, it causes pain relief. And we took that idea and, as I mentioned, one of the people who's actually now working part-time for the company as well, discovered USB5 is also the upregulation or the increasing in 3.2 channels in chronic pain is due to the overexpression of the enzyme USB5, and that this is because the USB5 enzyme associates with the 3.2 channel to increase its stability, so it prevents the normal breakdown and recycling in normal physiological conditions. Ion channels go to the membrane and they eventually break down and recycle. And under normal conditions, there's a certain amount of channels. In the pain condition, what happens is that the channels increase and transfer to the membrane so that there's more channels in the cell membrane. And the more channels that accumulate, then the more electrical activity and the more pain signal happens. And so this is caused by ubiquitination being halted. So ubiquitination is the sort of normal recycling of proteins. And what the DUB ubiquitin enzyme called USB5 does is prevent that recycling. So the channels keep accumulating and so that there's more and more. And this creates the chronic pain signal state. And so what we wanted to do, or what we realized, is that if we could prevent the DUB ubiquitin enzyme from interacting with the channel, it will allow that normal recycling process to occur so that the channel will come back down to its normal levels of physiological function. So that's the beauty of our technology, is that we're not actually directly inhibiting either of the proteins. We're just inhibiting their interactions so that those two proteins continue to do what they normally do. They just don't like they do under chronic pain state.
Yes, calcium, of course, is very important in many different cell functions, from release of neurotransmitters to muscle cell contractions to this particular calcium channel. I'm assuming it's in nerve cells, it's not in any of the supporting cells. What is it? Again, you can correct me. What is its role?
3.2 is actually found throughout the body. It's in neuronal cells as well as in the peripheral nervous system as well as the central nervous system. Its primary role in the human body is it's the sort of trigger mechanism for electrical signaling, especially in the heart and the brain. It controls the rhythmicity. So, you have a regular heartbeat or you have regular brain signaling. A 3.2 channel is thought to be the sort of mechanism that regulates the sort of continuous electrical pulsing of both the heart and the brain. So it's dysregulated. 3.2 can actually cause arrhythmia and can also cause diseases of the brain where you get asynchronous activity. For example, epilepsy is the one that I mentioned earlier. Cat3.2 is being implicated in epilepsy. It's also being implicated in arrhythmias. Mutations of the channel can cause disease states. So it's very important. That's its role. Pain has a different role. It's found throughout the pain signaling process from the cells that detect in the nerve endings the site of injury, or right the way up through the, we call the dorsal root ganglia, to the spinal cord, and up through the spinal cord signaling processing part of the brain, finding something that can inhibit only in a chronic pain state is ideal, rather than what a lot of drugs do, which is directly inhibit ion channels and directly inhibit the channel, because that could cause other effects on channels that are not involved in the pain processing.
First of all, it sounds like under normal circumstances, in the brain at least, the calcium channel almost acts like a pacemaker, and it creates different waves that you see on EEG. There is increase in these calcium channels in a lot of chronic pain states, I'm assuming based on animal studies. And the increase isn't just in the brain and isn't just in the spinal cord or isn't just in the nerves and also root ganglia that go to the tissues of the body. The increase is actually at all the levels.
Yeah, we've done most of our studies show the accumulation in the peripheral nervous system in sort of DRG spinal cord to show an increase. And again, this is where we feel the USB5 is interacting. We see elevated levels of USB5 in chronic pain. We believe this elevated levels of USB5 as a biomarker for chronic pain, as pain is a very subjective feeling, and so it's very hard to measure quantitatively. And so we feel this might be a way, at least measure in some way a person's level of USB5 correspond with their level of chronic pain, and therefore create a more targeted approach for chronic pain patients. But that's a part of the area of our research that's a lot more preliminary. We focused mostly on developing drugs that sort of inhibit the interaction so that we reduce the levels 3.2 chronic pain.
So it sounds like you have your primary angle, and that is to develop a treatment for chronic pain, but also you have a secondary almost endpoint where you could potentially develop a diagnostic tool that could be used in chronic pain management. But one advantage of this approach is that in chronic pain conditions, these calcium channels, 3.2 type calcium channels, they accumulate. There's more of them than is found under normal circumstances. And the reason for that accumulation is not that cells which transmit pain signals make more of them. The reason for this accumulation is that they are not broken down normally. And your approach is to basically make it so that they are broken down normally again. So it will only affect those cells that have excess number of these calcium channels, but this approach will not affect cells that have normal number of calcium channels. Is that right?
Well, that's exactly right. And that is what we think is the sort of primary differentiator or advantage of our technology, is that this mechanism only occurs in chronic pain states, so that it allows us to target channels that are accumulating in a chronic pain patient. That's what we think is a distinct advantage to our technology. It will also decrease the likelihood of side effects, which a lot of drugs have by directly blocking their protein target. Our mechanism is only found in a chronic pain state. So it will be able to far more discreetly target chronic pain and also not affect those proteins' normal physiological functions.
There's multiple subcategories of chronic pain, as there's neuropathic chronic pain, there's mechanical chronic pain, there's even chronic pain originating from internal organs. What type of chronic pain is this approach targeting?
It's a good question, and our research data shows that our drugs work on a wide variety of chronic pain models that we have in our lab, from diabetic neuropathy induced pain to acute induced pain, as well as inflammation is another one that we're able to have very good animal models. Of course, we are still in the preclinical stage to obviously can't test on humans yet and see which specific chronic pain diseases that they'll be most effective of in humans. That's obviously where we're aiming for and trying to get to. But it seems to work on a variety of different chronic pains. It's not just the one.
I'm not sure how far you've gone with this research, but is it a small molecule that you can swallow? Or is it more like a protein or an antibody that has to be injected?
It's a small organic molecule which we have tested in various animal models, and we've administered it in a variety of different ways, but also oral dosing the animal. We're just in the beginning stages now of testing PK pharmacokinetic profiles of the drugs as to how stable they are and how easily the bioavailability and distribution of drug is in the animals. For that, we're using independent companies to measure this, how the drug is distributed and excreted, how long it stays in the system. And those results will determine the dosage. Our goal is to try and produce a one a day orally available pill that'll be effective for 12 hours or more. And that's the target. But the results will guide how that works. And we have several lead compounds right now that we're developing, and we'll go forward with the best one that most closely hits those different targets. But we are aiming for something that's going to be orally available.
Does the medication need to penetrate blood-brain barrier?
That's the indication that's happening by oral administration, that it works. So I would suggest it does. But that is part of what we're testing right now, or we've sent our compounds to be tested to a CRO to test exactly those things, how the drug is distributed and how long it stays in the bloodstream and system, how quickly it's broken down, excreted, and so on and so forth.
Now, you mentioned animal studies, and I guess there's animal studies, there's typically human studies, and then there's another methodology that I guess was developed in the last maybe 10, 15 years, and that's organ on a chip or organoids, where you can actually take human cells and it behaves a little bit more like a human liver or human heart or human brain, and test medications on that instead of animals, with the idea that perhaps I suppose it's more efficient theoretically, and because you don't have to store animals in cages and all of that sort of stuff. And B, presumably the results would be closer to what you'd see in human trials because these are human tissues. I don't know if you have any experience with that.
I think it's definitely worthwhile. The more results you gather, the better. We have sent our drugs to be independently tested to a company that does cardiac safety profiling for drugs, which is, of course, a very important first safety step. And what their system is, they've developed to differentiate human cardiomyocytes. And so those are human cardiomyocytes that they basically grow in a dish, and then they basically squirt our drugs onto those cardiomyocytes and see how they react to the drug, whether it changes the action potential or the sort of rhythmicity. And I'm pleased to say that our drugs came through with flying colors, so they did not have any impact on those human cardiomyocyte cells. And that's definitely a good result. But getting the results back from live human cardiomyocytes gives us the confidence that the drugs are safe from cardiac point of view. So I think those are very interesting. I'm not an expert on organoid development. It's definitely growing. And I believe we're at the stage now where a lot of this testing, I mentioned the safety, toxicity, and pharmacokinetics, is all done independently now by CROs, and they are the ones who do the testing. And whether they use animals or organoids, the results will tell us what they do. And there are requirements from the FDA how our drugs will be. It has to meet those requirements. So I have no idea if all of the organoid testing meets those requirements yet. So you want to be as close to human conditions as possible before, of course, administering the drug to human.
I guess a couple of things I'm picking up from here. One is regulation. This is a very regulated field, and a lot of what you do is dictated by what FDA or Health Canada says is required before you can proceed to the next steps. So it's not just about what works best, but also what actually meets the regulation, because even if something can work better, you're not going to do it if it doesn't meet the regulation that allows you to move on to the next step. And which brings me to the next question, and that is, how far away are you guys from studies in humans?
That's It's a good question because it depends a little bit on how quickly we can get the financing done to sort of complete all this safety testing, the regulatory safety, so that we can meet the investigational new drug designation from the FDA. So we are in what is called the IND phase, the investigational new drug enabling phase, where we have to do all the safety and toxicology and pharmacokinetic testings. So that requires a lot of money to get there because all these tests have to be independently done by CROs. If we get the money, we believe that we can these results and apply for IND designation within probably the next 18 months. And once we get the designation, and part of that designation is to come up with a plan for phase 1 human clinical trials. But you can't go into phase 1 until you have all of the other safety tests done and get your IND designation so that you can begin to do that. And then, of course, it takes quite a lot of money to perform a human trial as well. In a perfect scenario, if we can get IND designation, we could potentially be doing human trials 2 to 3 years. But it all depends on the finance. Hopefully, we don't hit any roadblocks.
Yeah, startups, of course, are very much affected or limited by the funding.
Yeah.
Developments inside a big pharmaceutical company, startups much more sensitive to funding. So I guess assuming everything goes well and you end up having a human trial and the human trial is successful, I don't know that I'm aware of any Canadian-based pharmaceutical company that I guess stayed in Canada, headquartered in Canada, and is, successful.
Mm-hmm.
I've seen some companies that get bought out by US or European pharmaceutical companies once they reach a certain stage, but I've never really seen one that's actually stayed independent and ended up headquartered in Canada. Any thoughts?
I have lots of thoughts on that. So the reality of especially drug development business, in particular the field that we're in, which is pain research, the vast majority of the money and funding is in big pharma, and a lot of that big pharma is in the US. For ourselves, I mentioned we need financing to get through the IND phase. We also are going to need financing to get to the phase 1 clinical trials. We're talking tens of millions of dollars. And it's that kind of money isn't really available in Canada. It's very hard. And so we realistically, if we want to see our technology to progress, we have to at some point either license or partner with a big pharma in the US or somewhere else. The US obviously would be the most obvious choice because it's close. If the end goal is to develop a drug that's actually going to be effective and make it to market and it's going to help millions of people with chronic pain, it takes hundreds of millions of dollars. And for a startup, that's not realistic. Even if you are able to stay independent from a big pharma, money that would be needed to raise to do that is more likely going to come from the US as well. I think we're realistic enough to know that with the amount of money involved and the logistics involved as well, have taken a drug all the way to market. It takes a company with deep resources to do that. For startups, it's not practical or realistic. In our field, at least we'll find the money. They call it the valley of death, what we're going through, because it's so difficult to find financing for the preclinical stage. Once you get into human trials, then more people are interested in funding it, but then your technology is much more mature then, so the risk is less. We do get certain amount of backing from governments, and we get certain amount of funding and backing from the University of Calgary too, but the kind of money that we need in order to succeed far exceeds what they can offer us.
So we'll end here. Thank you, Dr. Bladen, for taking the time to speak to me, and best wishes for your startup. Certainly, we need more pain medications We have a very limited number, and most of them have been around for about 20 years. I just think it's wonderful that more people are trying to develop new treatment options for chronic pain. Thank you.
And thanks for having us.
So, what are my final thoughts about Zymedyne? Obviously, the market for a new chronic pain medication is huge, as 20% of population have chronic pain, and the medicine has a potential to be a breakthrough in the field. It doesn't just act on the spinal cord, but potentially can act at other parts of the nervous system involved in processing pain signals. So far, it appears to be safe. However, the question is, will this company succeed? And if it does, will it stay in Canada or move to the US? Historically, a lot of great medical discoveries have been made in Canada. That includes discovery of stem cells, discovery of insulin, even discovery of the chemical pathways that led to development of Ozempic. However, commercialization of these discoveries happened in other countries. And the windfall that accompanied these discoveries again stayed with countries who are good at commercialization, not necessarily countries who just discover new medical facts. So Canada is very good at funding basic research and nurturing scientists and allowing them to pursue sometimes topics that are not popular. It is also arguably pretty good at funding startups at the point when they are created. What it fails at, however, is growth of these startups. Typically, companies in Canada can get access to money in tens of thousands or even hundreds of thousands range. But as you heard during the interview, for a company to produce a new drug, it will likely take $150 million just to get to that point that it becomes sufficiently far along that other outside sources will invest significant sums of money. There is a statistic that it takes about $1 billion to bring a drug to the market. So in my mind, the question is, how can we make it so that Canadian discoveries become commercialized in Canada and we create companies that stay in Canada? Well, unfortunately, I don't have a simple answer for that. However, I have some possible thoughts for solution. Uh, Perhaps instead of providing small grants to multiple startups, we should develop an area of expertise so that we can provide much bigger grants, but to fewer startups. As you heard during the interview, smaller countries have less money than big wealthy countries like US or China, and they cannot compete financially with those countries on everything. However, if they focus on a specific angle or specific part of the medical field, perhaps they can outcompete bigger countries in that small field. Another possible solution to complement my first idea is to create an environment where we can attract major American and possibly Chinese VCs. Governments are pretty good at funding basic research and at providing money to very early startups, but they are notoriously bad when it comes to growing a company. And I think part of the problem is that government administrators are not sufficiently incentivized to make sure that the companies they're funding succeed. In partners in VC firms, VC stands for venture capital, on the other hand, live and die on the basis of success of companies they invest in. If every single company they invest in goes bust, the VC firm will be out of business. So they're incredibly incentivized to not just invest money in companies, but also to make sure these companies succeed. A lot, but not all VC firms, in addition to providing money, will have a seat on the board where they will often mentor first-time founders of startups, as well as open the doors for them, as well as helping with recruitment of qualified employees. So VCs don't just bring money and appetite for high risk, high return outcomes, they bring smart money, which actually increases the chances for the company succeeding. Finally, the government would have to create some kind of a carrot and stick tax environment which would encourage VCs not to move the company to the US once it reaches a certain size, but rather leave it in Canada. For too long, we have been too dependent on the US. For too long, we have also been very dependent on our natural resources. And while US economy is primarily driven by tech, our economy is still driven by the same industries that drove it in 1950s. I think it is time for a change. Thank you.
Disclaimer: when it comes to your health, always consult with your own physician or healthcare provider for personalized advice and guidance. The information provided in this podcast is for educational and informational purposes only and should not be considered medical advice or a substitute for professional medical care. Be sure to follow our Instagram at Karmy Chronic Pain for updates on new episodes and more educational content today.